Well, good afternoon, everyone. On behalf of the College of Engineering, and in fact also on behalf of Biomedical Engineering, since we are in their home turf here, I'd like to welcome all of you to the College of Engineering Distinguished Lecture, and it's a real treat. Today we have a very special person. So our speaker, Dr. Kristi Anseth, who is a distinguished professor and the Tisone professor in the Department of Chemical and Biological Engineering at the University of Colorado, Boulder. Dr. Anseth's talk today is entitled Materials to Study Cell Biology in the Fourth Dimension. She is considered to be the pioneer in the field of tissue engineering and as a designer for new biomaterials for applications in drug delivery and regenerative medicine. Her work is unique. It blends together polymer chemistry, physics, molecular and cell biology, and molecular simulations and modeling. Her primary research goal is to pioneer the development of biomaterial systems for 3D cell culture to further the fundamental understanding of how cells receive and exchange information with their extracellular environment, and then to use this knowledge to engineer material niches for translational applications in cell delivery and regenerative medicine. Her seminal work indeed has revolutionized the field. Dr. Anseth was elected to the National Academy of Engineering in 2009 and to the Institute of Medicine of the National Academies in 2010, becoming the youngest member ever at both institutions. On April 26 this year, she added the missing academy, and that is the National Academy of Science, achieving the rare acknowledgement of being inducted to all three academies. Popular Science magazine named her one of its brilliant 10 in 2008. In the same year, the American Institute of Chemical Engineers cited her among its 100 chemical engineers of the modern era. In the early 2012, she was inducted into the Colorado Women's Hall of Fame alongside acclaimed individuals such as Temple Grandin and Colorado Supreme Court Justice Mary J. Malarkey. Dr. Anseth grew in northwestern North Dakota. Serious about basketball from a young age, she played both basketball and volleyball after enrolling at Williamson State College, where she earned the honor of academic All-American during her sophomore year. See all the things we can find in the web, Kristi? She then transferred to Purdue University, where she obtained her undergraduate degree in chemical engineering in '92. And this was followed by 2 short years by a doctorate in the same field at University of Colorado, Boulder. She served as a research fellow at MIT before returning to her Colorado alma mater in '96 as an assistant professor of chemical engineering. She also became an associate professor of surgery at the University of Colorado Health Sciences Center in Denver, a position she holds to this day. In 2000, she was named a Howard Hughes Medical Institute assistant investigator, the first engineer in the U.S. to be so honored, and she graduated to a full investigator in 2005. As a faculty scientist, Dr. Anseth spends a great deal of time teaching both undergraduates and graduates in the classroom and in her laboratory. In fact, her devotion to classroom give and take has had to do much with her decision to remain in academia. She cherishes working with students, and her instructive creativity has been recognized by several teaching awards, including the 2008 Distinguished Engineering Alumni Award for Education and UCB's 2013 Hazel Barnes Prize, the university's highest faculty recognition for teaching and research. I'm particularly inspired by this quote, and I quote Kristi, "Beyond teaching the basic scientific method," she says, "I try to get my students to take risks and to explore the unexpected. This is where some of the most interesting results lie." Join us now as we explore the unexpected with Dr. Anseth. It's my great pleasure to welcome her to Davis. Join me in giving her a warm welcome.
Well, thank you for the kind introduction and for the opportunity to visit UC Davis. This is my first time on campus, but of course I've met many of the faculty here and followed the research with great interest. There are lots of wonderful things going on. And what I'd like to share with you today is walk a little bit through some of our interests in this area of how you can design materials, ways to culture cells in multiple dimensional space, and learn something about the biology of how those materials can guide and direct cells with the hopes and applications in regenerative medicine or tissue engineering applications. So I'm going to give a little bit of background and walk through where we started in this journey about 15 years ago, and then hopefully by the end of this talk tell you about where some of our current thinking is going. So the idea is that we're working in this space of the microenvironment, the material system that surrounds a cell. And cells get lots of different information from this environment. And if we can design different types of biomaterials, or cell scaffolds, if you will, that one could culture and expand and these cells in 3 dimensions, this could have applications in regenerating and growing tissues. And my first story will be a little bit about cartilage, because I know there's lots of interest and expertise here in cartilage tissue. But many times, we also need to take a step back and think about, as we look at different types of tissues and some of the bottlenecks in designing systems that may have a more complex function or multiple types of nerves and muscles and blood vessels. Oftentimes, we need to figure out which signals do I need to present to the cells. And oftentimes, we don't understand how cells interpret those signals. So that brings us to some of the basic biological questions so that we can begin to put together and engineer systems that would allow us to manipulate cells by proper design of that material microenvironment. Now, there are lots of different types of biomaterials that one can select when culturing cells in 3 dimensions. And one of the types of materials that my own group has had a lot of interest in are hydrogel materials. So here's just a macroscopic picture of a hydrogel. If you could see the molecular level details of that hydrogel, what you would find would be long macromolecular chains. And these long chains would normally just dissolve in water. But because of interactions between those chains, it renders them insoluble. And instead, they behave more like a solid or a gel that imbibes large amounts of fluid or water. So the idea then is when one embeds cells in this type of environment, it imparts many properties that are found in numerous soft tissues in our body. It imparts many of the tissue mechanics and our elasticity. The high water content allows for transport of important molecules that you might add to the media when you're culturing cells, or molecules that the cells are secreting and communicating to one another. So why is an engineer interested in these types of problems? Because oftentimes, when you go and look at figuring out these signals, many times it's looking at processes on different size scales and on different time scales. So from a cell at the nanometer level binding to and interacting and receiving a signal from this environment, to perhaps it extending a process or moving size scales of tens to hundreds of nanometers, to all the way up to something that might be a functional tissue subunit that could be several millimeters. As well as on time scales, what happens the instant that I assemble all this, as a cell begins to interact and remodel that, to several weeks later where you may begin to have something that begins to look like a deposited tissue. So one of the ways that we think about these hydrogel systems and trying to figure out some of these signals is we think about how simple is complex enough. So what do I mean by that? Well, on the one hand, we can think about these hydrogels as just being a permissive environment. So the cell's not really getting any type of chemical or biological signal from this environment. Instead, it's sensing its position in space, perhaps the mechanics or physical properties of that surrounding environment. But the cells are doing all the work to secrete and deposit and make the biological microenvironment. So that we call permissive. But on the other hand, permissive is too simple. It's not complex enough. And sometimes we need to coax and put in specific biological cues that will promote a desired function. promote a stem cell to become a certain cell type, like a cartilage cell or a bone cell. And many times that promoting then involves incorporating different types of biochemical cues. So I'm going to walk you through some examples where we began through some of this permissive space, which then led us down a path of needing to have more promoting environments. And we're going to start with the applications, and then we're going to end with some of the examples of how you can use these materials to get at basic biology. So the area that I want to start with is in regenerating cartilage, trying to treat arthritis. Many of you probably know somebody who's had a total joint replacement. Cartilage is a tissue that's found at the ends of our articulating joints, such as this knee joint here. But it's one of the tissues in our body that has a very limited capacity for self-repair. It's mostly an avascular tissue. It's not innervated. And when cartilage becomes damaged and wears, what happens is this nice property that it has of cushioning and lubricating our joints so that there's not pain upon movement. When it deteriorates, you can get bone on bone, and then it becomes very painful. And it's a progressive type, of course, and that leads to almost 250,000 total knee replacements performed in the US each year. So we have some collaborations with orthopedic surgeons that I began early in my career. It's a collaboration with another bioengineer, Jennifer Alicieff, and orthopedic surgeon, James Wentz at Hopkins. And this was a time when there were beginning to be different types of cell-based therapies to treat cartilage regeneration. So one of the patients that the orthopedic surgeon was seeing was a 41-year-old woman who injured herself playing soccer in high school, and now it's degenerated to the extent that she has debilitating pain. So anybody that doesn't want to see this, this next picture is an open surgery. So you can put your head down and I'll tell you when the picture's off, but here it comes. So here's the knee joint. Here's what healthy articular cartilage looks like, and here's the very large defect. Okay, so it's off. So in the 90s, in the mid-90s, I was beginning my faculty career and thinking about designing cell delivery systems for tissue regeneration. And at this time, Genzyme was marketing its autologous chondrocyte implantation technique, where they would take a biopsy of cartilage from a non-weight-bearing region in the patient. They'd digest that tissue and get the chondrocytes, the cells found in cartilage. And then they'd expand this. And then once they'd expanded the cells, deliver it back to the patient through an injection. And so the idea was that the patient has lots of healthy chondrocytes. Can we coax those cells to repair the tissue where it's damaged in the joint? And so this is just a region where they take these biopsies and what they look like. And then I'm going to show you one more open surgery. So here it comes, if anyone doesn't want to see. So here was the procedure. They'd take periosteum that surrounds the bone and suture this in place over the tissue, and the cells would be injected and be underneath here to kind of keep the cells in place. All right, so that picture's away. And the problem, though, with the procedure was that depending upon the type of defect and the surgeon, results were highly variable. And it seemed to work well for small focal defects in cartilage, but the large defects, like the ones that I showed you, it didn't work very well at all. And unfortunately, that's when many patients go to their orthopedic surgeon because their joint is very painful. And so the idea was, in our minds, if you look at cartilage and articular cartilage, we thought this might be a good target for us to start in that there was an obvious cell source and there was already a procedure in place of getting the patient's own cells. And these cells, if you take a piece of cartilage and you section it, and a thin section, and then stain it, you can kind of see these cells here, the chondrocytes. And the primary job of those chondrocytes is to maintain the matrix and the tissue function. And so this blue is a stain for one of the matrix molecules that it makes, collagen. And it's one of the few tissues that it happens to be type 2 collagen that's predominant in the tissue. And then down here is another molecule, these proteoglycan molecules. These are large sulfated sugar molecules. And the idea here is the collagen, as a simplification, is sort of like the mechanical struts. And the proteoglycans allow the tissue to imbibe large amounts of water, and these combined give it some of its really unique properties. So our idea was, well, the tissue has a large amount of water. Could I design just a hydrogel carrier system that could be used, co-delivered along with the chondrocytes, could be injected into the defect, and then it would solidify in situ? And we were interested in using light to solidify this, since light's very compatible at being able to make gels without having to heat them up, expose them to any harsh type of chemicals. Instead, it just can be done at physiological conditions in situ. And that the gel would form, and then over time, the cells inside of the hydrogel would hopefully regenerate a cartilage surface, a functional cartilage surface. So here we were thinking from the translational standpoint, what types of materials should I use for this? And we were using a couple of components to make these materials. And the first was we used polyethylene glycol, PEGs. So very hydrophilic molecule, widely used in human medicine. It's used to conjugate to lots of proteins to improve their lifetime and solubility. So we chose PEG as our core of the material. And then we grafted off of that lactides. These are what degradable sutures are made of. And so these were going to break down at physiological pH and hopefully in a manner that would match the rate at which the cells would deposit their matrix. And then we put on different types of N groups, here acrylates or methacrylates. The idea here is that these N groups would transition these soluble polymer chains to a solid gel through a reaction through those reactive acrylates and methacrylates. And we were going to use a light catalyzed reaction to do that. And indeed, you can embed chondrocytes inside of these hydrogels. For those of you in the audience working on proposals or more junior to the field, all of my first NIH proposals that I wrote, everyone said this is, they thought it was a bad idea, that cells would never survive this photoencapsulation process, and that just wouldn't work. And so we had to persist, and indeed, some of you might know of photodynamic therapy, which is using light to kill cells. But there are conditions that you can engineer and control, and it's a broad range that you can physically encapsulate cells and proteins and do these reactions in the presence of tissues. So once we could do that, one of our first projects that we were involved with was then asking questions. Okay, so I can control these hydrogels, and one of the things that you can control about it is how many of these links exist between the chains, sort of a three-dimensional mesh or a net in space. And that links to many of the properties of the gel that you make. So on the one hand, we wanted to have high water content so that these cells could easily have diffusion of the molecules they're secreting. So that was high water content was one thing we wanted to have. So you'd want to have a very open gel structure. But on the other hand, it needs to restore mechanical function or structural integrity to a damaged joint. So you have to also worry about the properties, the mechanical properties of the tissue, and that would take us in a direction of having more links to give more robustness to the equilibrium modulus of the gel. And it's a trade-off between these two types of properties, and also other things, you know, like this diffusion of molecules also depends upon this mesh size or cross-linking. So we went through a systematic study of studying many different cross-linking densities, and we looked at how the cells would respond to this and conditions where they would survive and where they could deposit these matrix molecules. And it turns out if you look at one of those matrix molecules, the proteoglycans, you can see that there are regions here. So now here, the dark spheres here, those are the cells, the chondrocytes, and they're secreting and making these proteoglycans. But in this case, diffusion is more limited. It's hindered, and you have regions where it's largely gel, and you get heavy staining around the cells only in certain regions. But if you open up the mesh size and you change the diffusivity, you can have facile transport of that molecule throughout the gel. And we like to use models to understand this, and we know that the mesh size of our gel and diffusion coefficient of a reasonable size proteoglycan, we can model cells at different density and sort of predict what would happen with these distributions and understand that as we increase the mesh size, we change the diffusion coefficient, we can have a system that becomes more uniform. But the problem is that this is just one of the components, the proteoglycans, but collagen is a very important component to the mechanical integrity of the engineered cartilage. And when you look at collagen, it's a very complex assembly to create these collagen fibers. Cells secrete many pro-collagen molecules that need to assemble, and the fibers that they assemble into can be hundreds of nanometers in diameter. And so even when you look at this system, the collagen is all stained right around and residing in the lacunae of the cell. So what's happening here is that for these large-sized molecules to assemble, you need to have breakdown and erosion of your hydrogel. And so you need to have that fourth dimensional control, not just of the structure of the material, but how it changes with time. And I won't go into the details of all of this, but we did some modeling of this system as well to understand the kinetics of how those crosslinks would hydrolyze with time. And we measured lots of properties and their changes with times. An evaluation of those gave us time constants for the kinetics of the hydrolysis. And then the connectivity of our network, how many links existed between the chain, would dictate how the gels would lose mass. How many links did I need to cleave, and what rate did I cleave them allowed us to predict things, how these gels would degrade. And early on, they start to erode mass and lose these crosslinks and open up. And then at a later stage, what happens is it transitions from being an infinitely cross-linked mesh to something that's now just highly branched and soluble again. So you get this critical point where the gel will completely dissolve. And this happens to be a really critical region to get a lot of the collagen to assemble. And we did lots of experiments looking at systems where here, the gel's degrading and eroding at a rate that hinders collagen matrix deposition. It's all in the lacunae. Whereas here, the gel reaches this point too quickly, and the cells haven't secreted enough matrix, and you get large defects or holes as the gel solubilizes. And here, you can find conditions, depending upon the cells you're using and the density they have, where you can get pretty uniform matrix production. So I had a couple of my first PhD students work on this aspect of the problem, and then we collaborated with clinicians to look at various types of animal models and translating this to see if we could create different types of defects in animal joints and be able to repair them by placing our gels in situ and following the tissue production with time. And what I'll say is that we could do a pretty good job of regenerating cartilage, and we could follow some of these animals for up to a year. But some of the things weren't necessarily so simple as we thought. So here's a section of what some native cartilage looks like, and here's a typical example of what we could kind of regenerate. And it's not just about producing matrix, but it's that organization of the matrix that's very important. And as it opposes the bone here and what happens at the surface for the lubrication. And we get some of those aspects, but our simple permissive matrix perhaps doesn't give quite enough signals to guide this. And in fact, to this day, there's still major problems with regenerating cartilage in terms of getting it to integrate to the surrounding tissue, maintaining its articular cartilage phenotype for long periods of time, getting enough collagen produced and elaborated. And so on the one hand, I think we've made lots of great strides in this area. It's a tissue that even something that we thought was fairly simple, one cell type, healthy cell source, and that we could design systems to culture them and coax them to form tissues, wasn't complex enough. And we still have to try to solve this problem for the field. And so then that made us begin to think about, well, what could be some alternative strategies, and how do we take these simple gels that we were working with, engineered to degrade at certain rates, and think about systems that could be more promoting. Perhaps give cues so that our cells might signal to surrounding tissue, or perhaps deliver cues, and think about what those important types of cues might be. And so for that, we began to rethink some of our hydrogel design because these basic permissive gels, what they're really good at is you can embed cells in 3 dimensions and control and engineer physical properties and degradation properties tunably, but the cells don't recognize any of that chemistry. So on the one hand, they can be good for culturing cells like chondrocytes that have a natural rounded morphology, or if you want to promote cell-cell interactions and create different types of cell aggregates. But many, many cells need to interact with their surrounding matrix environment, and they get very important cues. As one example, here are some bone marrow-derived stem cells, mesenchymal stem cells, and if you put these cells inside of these permissive environments, what happens is they want to attach and bind to their matrix, but they don't recognize that chemistry. These cells want to move towards one another, but they can't because the engineering of the degradation is at a much slower time scale. And in fact, these cells get very frustrated after a few days in the matrix, and they'll undergo a programmed cell death, an anoecus, because of lack of matrix interactions, lack of signals from their matrix. So we began to think about, well, on the one hand, what is naturally found in a cell's material environment? So cells are surrounded by a proteinaceous matrix of collagens and laminins, and sometimes these proteoglycans and elastins, and they're getting all types of signals from this. So you could say, well, why don't you just take the native extracellular matrix, reassemble it around the cell, and that would be the perfect world. You'd have a tissue all put together. The tricky part, though, is a lot of times those proteins can be very difficult to manufacture and make in a reproducible way, in a way that they don't denature. And so we began to think of complementary strategies to that approach and think about, well, minimally, what is it that's found in the extracellular environment that I'd like to capture in a synthetic environment in a simplified way? And so our permissive hydrogels had high water content for good transport and could give mechanical cues. But what we were missing is we were very interested in signals for cells to bind to that adhesion proteins naturally do in the matrix. In a native tissue environment, cells also secrete proteases and enzymes that can degrade their matrix, and that's really important in natural wound healing. It's also a reservoir, storage depot for numerous growth factors that are released on demand, and that also allows tissue repair and regeneration. So we were interested in, well, how could I take my PEG hydrogel environment and make it more promoting? And the approach that we took, and we were learning in the literature at that time, was how from different proteins there are specific regions, small peptide units, that can recapitulate some aspects of those full-length proteins. So for example, in the adhesion proteins, we know regions exactly where cells bind. RGD is found in fibronectin, IKVAV is found in laminin, and we can make these synthetically in the lab very reproducibly using solid phase peptide synthesis. We know different sequences where proteases that cells secrete will degrade, and here's one found in collagen. We can also screen through all different types of peptides and the way that they'll bind molecules with affinity. So here's a short sequence that binds the cytokine transforming growth factor beta with high affinity. And so, this was at a time in the field where there was lots of interest in using peptides as these synthetic mimics of proteins. And Jeff Hubble and Jennifer West and others were beginning to look at these systems. And so what we did was we were really interested in using a particular reaction or chemistry to incorporate these peptides in an easy manner. And the chemistry was this. We wanted to use cysteine as one of our amino acids in our peptide sequence because cysteine has a thiol group. So if I can make peptides of any sequence of interest and include cysteine, this is going to be my reactive handle for cross-linking my gel. And then I'm going to use my PEG molecule, and we screened through lots of different complementary reaction components to this, and we came upon this norbornene that reacts very, very efficiently with this cysteine. But what's unique about this is you mix these two together, and they're completely stable, And it's only when you expose it to light and catalyze the reaction that this will occur. And so the idea, for those of you interested, we expose to light, it creates a thionyl radical on this cysteine, which propagates very efficiently through this ene, and it creates this carbon-based radical. And unlike my acrylates and methacrylates, this carbon-based radical cannot polymerize through this. It can only chain transfer back to my thiol, and it repeats itself. So it's a really unique reaction mechanism that's very nice for clicking together gels where I can use PEGs and peptides and incorporate them in a very efficient manner. Now, the nice thing about the light activation of this is, I'll have a little side, is that when you use light, you can also pattern chemistry. And I'll explain why this is important to us in the last parts of my talk. But in general, if you leave some of these excess enes inside of a hydrogel, and then you swell in any thiol-containing signal that you'd like, an amino acid or a protein, and if you pass it through a mask, you can react and change chemistry in a spatially defined manner. Or if you use a confocal microscope or a laser source, you can also write chemistry inside of gels at spatially defined points in 3 dimensions. It's a very fast reaction, and all this can be done in seconds, almost at the same speed that you'd use for just imaging cells. All right, so our idea, though, was we wanted to use our PEG component, our cross-linking density, to control physical properties of our network. And then we wanted to click in biological signals based upon the peptide sequence that we chose. And we would choose these peptides depending if we were working with neurons or if we were working with a stem cell population, depending upon our tissue of interest. And so now let me just show you one particular example where here we take a PEG that has 4 arms here, and we have these norbornene functionalities, and we're going to use a couple of sequences here. This is one that's degraded very efficiently by those bone marrow stem cells, and here's the RGD so that they can now attach to it. So when we put those cells inside of these PEG gels, rather than them anoecusing and dying, instead what happens is the cells attach and spread, and they're locally degrading this environment at a rate and decisions that the cells make. And if you change this tryptophan to an alanine so that the cells can't degrade it quite as quickly, then what happens is that the cells can survive because they can bind to the RGD, but they just can't move or very quickly throughout this matrix. So what we were thinking about was we wanted to design this type of system that we could load up with different types of signals so that only certain resident cells could migrate into this environment. And when they migrated, they would encounter that signal that would promote tissue regeneration locally. And the motivation for this also relates to some research of interest here as well in terms of finding factors to induce bone regeneration, osteoinduction. So one of the biggest successes in some of the tissue engineering field has been the discovery of different types of bone morphogenic genetic proteins. And they're all different types of factors that we're learning about and targets. But the problem is that you have to localize that signal. Like, even in the current products to deliver BMP for bone regeneration, they have to use a dose that's about 200 times what would be in all of the bone marrow of an individual. So it becomes very important that these signals stay local so you don't get off-target effects. And so we had a medical fellow rotating in our group from otolaryngology. So he's trying to regenerate bone in cranial defects and injuries. And so this happens to be an image of a rat skull, and we created a critical size defect. It's 9 millimeters across, and it won't heal on its own. And so you can imagine, especially cranial defects, that you want to repair and regenerate bone fast because it's protecting the brain. And so we used 2 different gels, and we loaded them up with different osteoinductive factors. Here's just one with a recombinant BMP. I call this one non-degradable. Non-degradable is the one that the cells couldn't infiltrate very quickly. And here's one that's the MMP degradable. And what's interesting about this is that, so you can see systems where we get some localized bone regeneration. Even though we have this potent factor here, it's only the cells that migrate in that will get the signal, and you don't get off-target effects of super excess bone form. And here we see large amounts of bone regenerated across this gap. So this isn't perfect. I think that what we see if we start to quantify some of these factors is it still takes 8 weeks. We don't completely close that defect. And this is only one signal that we were looking at. So now you can imagine, again, let's go back. How simple is complex enough? What else do we need to design into our system? Is it a dose that we need to change? Do we need to have more chemotaxis and get cells to infiltrate faster, to get bone to regenerate faster? Do we need multiple signals? And so this is where I'd like to motivate for the last part of my talk is, how do you figure out what signals you need to present to cells? And how can we screen through this in vitro? So in a more rapid way, I could learn something about the most important signals so that I could then go back and integrate this into my design of a more complex experiment that's in vivo or in animals or humans and improve products. All right, so the idea is we're going to design systems where we can change signals around a cell, and then hopefully by changing that signal, we can learn something about the process that we're trying to control. And I'm going to give this in the context of a nice collaboration that we had with a cell and molecular biologist. And so it started along this way, where we were interested in trying to understand and develop alternatives for treating heart valve disease. So in our hearts, we have 4 different valves. From an engineering perspective, they serve as these perfect one-way valves directing flow in our heart chambers. The tissue itself has, so this is one of the valves. It's an aortic valve, and the tissue is this thin little leaflet that's maybe 200 to 300 microns in thickness. Yet somehow this directs flow, opening and closing, 60 to 80 times a minute. Huge pressure drop across this. So what happens is that these cells inside of here have to constantly remake matrix and remodel this in response to any kind of injury that happens. But sometimes this process goes awry, and something happens, and rather than those cells maintaining this perfect functioning valve, they begin to deposit lots of matrix and make the leaflets very thick, and they don't open and close properly, and they'll even begin to calcify. And right now, there are no drugs known to reverse valve disease. You'd have a valve replacement, and this affects a certain large percentage of the population. So we had this collaboration where, so as a molecular cell developmental biology student, where she was interested in what happens to this one population of cells that it gets turned on this activated state and causes disease. And we knew that one of the main signals was this cytokine transforming growth factor beta. So Sharon wanted to say, what's different between these activated cells and the quiescent cells? And we first wanted to do a pan of all the different genes that would molecularly characterize those cells. But she did a control experiment first. And the control experiment she did was this, is she wanted to figure out what dose and time of TGF-beta to study. But first she just added one control where she took cells that she isolated from the valve and then immediately collected the DNA and did expression analysis. And then she took another population where she put on TCPS, and then she was going to dose them with TGF-beta. So tissue culture plastic wares, normally how we'd studied and looked at these valve cells. What happened, so I won't go into the details of this, but when you look at all the genes, there's a striking difference between the pattern of genes in the native tissue or a fresh isolate versus what happens if you just let it touch a plate for a few hours. And I want to put this in context for you. I won't go into the details of all the genes and their differences, which Sharon looked at, but thousands of genes go up when you put them on tissue culture plastic, and thousands of genes go down. And the problem is that TGF-beta, this potent cytokine that's really important for activating these cells, you treat them with TGF-beta, and after 8 hours or 24 hours, tens of genes go up, tens of genes go down, and the intersection between these is very, very small. So the message here is that the context with which you culture cells in when you're trying to understand these signals of what causes activation, let alone what reverses it, it's going to be completely masked by this huge background. And so the final thing that Sharon did was she then said, okay, let's look at what happens if you start to culture these in different environments, such as a gel environment versus tissue culture plasticware is about 6 orders of magnitude stiffer than valve tissue. And so she just put them on some of the gels that we were using, some of these degradable gel systems, and don't look at the details of the genes, just look at the patterns. Here's cells that you've taken out of the tissue and isolated their DNA. Here's one that you've never even taken them out of the tissue and you just extracted the DNA. Here's tissue culture plasticware. And here's just changing one thing, putting them in a hydrogel system. And the pattern isn't perfect, but it's much closer than tissue culture plasticware. So what I'm trying to motivate here is that context matters. And how can we learn how to make this better and have a better, more realistic in vivo environment for these cells? And how can I use that to study this activation and deactivation? So the gels that I've told you about today, we pre-engineered to degrade at some certain rate in a predictable manner. And that's not always great because you don't know what time scale you need. So the next step we took was we did these cell dictated. Let the cells decide when to degrade and locally remodel this environment. And that can be really great as a natural mimic of a native ECM environment. But you don't always know what the cells are doing to this environment. And sometimes what you'd like to do is to be an experimenter and watch my cells inside of gels and in these environments and trigger a change and watch how the cells respond to that change. And through those types of experiments, can we figure out how they respond to specific signals and in different manners so we can use it for improved bioscaffold design. So we had some people in our group working on ways to design materials where you could user-dictated change properties. So now we're still taking that same PEG molecule, but rather than putting on peptides or hydrolytically degradable groups, we link on this nitrobenzyl ether group. It's a molecule that undergoes a cleavage reaction when it's exposed to light. So it was widely used at the time for caging of molecules that would be delivered intracellularly and you'd expose to light and it would uncage. So we were interested in putting this in the extracellular space. And let me just show you a little bit about these. So you can take these cross-linking molecules and react them with all different types of, this is just a PEG monomer, and create hydrogel networks. But the difference with this hydrogel network is that inside of these linkers are photolabile molecules that when I expose to light, I can change the properties of the scaffold surrounding a cell. And so we wanted to first make sure that we could change this reproducibly. So we did some experiments where here's the gel, and we put it on a rheometer. And the rheometer measures the elasticity, the mechanical properties of the gel. And we can do this while we're simultaneously exposing to light of different wavelengths. And that modulus, or elasticity of the gel, is directly proportional to the number of crosslinks that I have intact inside of my gel. So if you do this, you can monitor the modulus, and it starts out high. And you expose it to light continuously. You're cleaving the crosslinks. And using particular intensities of lights and wavelengths, you can get a time constant for the cleavage. And if you half the light intensity, you double the time constant. And if you change the wavelength, you also change the time constant in a manner that's predictable just by how strongly that molecule absorbs light. And so this is just all photochemistry that we studied. And so these behave in a predictable way, depending inversely on light intensity and molar extinction coefficient. But so now here's the interesting experiment one can do. Can make materials with certain elastic modulus exposed to light, but rather than continuously, you shutter the light source, you remove it, and you've softened it. And it's at half of its original stiffness. And then you can re-expose to light and soften it again to a quarter, 20% of its stiffness. And it will stay at that value until you re-expose. So you can have a dynamically softening system. So the reason we were interested in this is that in our culture of these valve cells, I told you we were really interested in what makes them go from quiescent to activated. And the activated is important in tissue repair. But what happens when you culture them in supraphysiologically stiff environments such as TCPS is after about 24 to 48 hours, all the cells become activated myofibroblasts. You can never study them as a quiescent phenotype in this setting. And they'll even, when they're activated, they become highly contractile, and they snap and form these nodules. And these nodules will go on to calcify, express genes found in many osteoblasts. So this could be a model of studying the disease progression, perhaps. But we were really interested in, can I culture these cells in this state and then maybe begin to understand about some of this dynamic exchange with their environment, what causes activation and deactivation, with the goal, if I could understand that, maybe I could pursue different drug targets to reverse that when this population persists in heart valve disease. And so we used our photodegradable system. And the first thing that we wanted to answer is, are there regions where we can culture these cells where they're quiescent and regions where they're activated? And so we used these photolabile systems, and we passed a mask over this material to create a gradient in elasticity. So this leads to a gradient in light exposure. There's long light exposure times here, short. And so as a function of position, I have materials that I can measure with a nanoindenter what the modulus is as a function of position. And the red line is just what I would have predicted based upon exposure time from all of my rheological data. And then we can seed the VICs on these environments. And when you seed them, then the question is there a threshold with which they become activated? And is there a region where they're mostly quiescent? And so we did that. And one of the hallmarks of this activation of VICs is a particular protein, alpha-smooth muscle actin. So that's the only one I'm going to show you today, but there are other markers of this. And the red here is now the cell's cytoskeleton. The blue is the nucleus. And as you see, as you go to regions of higher elasticity, we see more activation. You can quantify this. And after 3 days, you have culture environments where you have almost no activated myofibroblasts. And you have very high levels of activated myofibroblasts. So now we have this culture system that we can better characterize these cells. And now the question is also, what happens to myofibroblasts? In the field, it wasn't really known. After they become activated, if they don't persist in the tissue, do they apoptose? Do they senesce? Do they revert back to the fibroblasts? And can you revert them back in a way? And so we did that experiment where we cultured cells under conditions that would cause activation. And then we wanted to say, well, what happens if once they're activated, if you could switch it to the environment where it should be deactivated? And so that's the experiment. You culture the cells in activated environments, and they become activated, evidenced by alpha-smooth muscle actin. Now you take half of your samples here, and you soften it in situ to see what happens to those cells. And you can watch this in real time. And indeed, what happens is it's reversible. They'll deactivate. They don't apoptose. And they behave as if they've always been in a soft environment. So now you can imagine we're doing all kinds of experiments about what happens. Is there a certain length of time? If cells stay activated too long, will it not become reversible? Or what happens if you go through multiple cycles of stiffening and softening? Will there become a point that it's not reversible? And we're learning how do the cells sense these signals so that we might be able to develop small molecules that could inhibit those cells and make them think that they're in a softer environment, so then perhaps they'd remodel and reverse some of the effects of valve disease. So just in summary, I'll end with a couple of comments. So I showed you just some different examples walking through increasing complexity. We still think as an engineer trying to figure out what are some of the key things to control rather than trying to recapitulate all the events that happen during development or tissue regeneration and wound healing. But this brings us to conditions where it's oftentimes not going to be about one signal, one protein, simply about stiffness, but perhaps we want to introduce a signal and then remove it and then bring in a second signal. Or perhaps we want to introduce a signal and let a cell degrade a matrix to encounter a signal that's being secreted from another molecule, another cell farther away, and then remove that interaction. And so towards that, you can begin to think about systems that we can learn about and control multiple pathways. And so here's one where we're culturing these MSCs in one of these degradable thiolene materials. But now we can pattern in regions and introduce a signal at one point in time, perhaps trying to expand or proliferate cells in a certain region. And then introduce a second signal. Maybe I want to differentiate the cells down a particular pathway. And I can create in a simple environment, just in this one example, 4 different regions of no signal, the red signal, the green signal, and both signals. And then if I combine this with my photolabile chemistry, now I can say, I'm really interested in these cells here. I'm observing something that they're expressing in real time under the microscope, and now I want to capture those cells. I'm going to erode them away. So that's one of the things oftentimes we don't talk about, is how biological experiments become more complex in 3D. We need ways to simplify that as well. And now maybe I would like these cells and capture those at a later point in time. And then I can do different types of these. I could deliver these cells to a patient. I could expand them in another environment. I could do more detailed characterization. Combining different types of approaches also afford opportunities where we begin to think about not just a single cell type, but perhaps multiple cell types, and trying to control their interactions. So we're also interested in ways that you might be able to study and get a motor neuron to extend a process that could form a functional junction with a muscle and get it innervated. It's a big problem. So we're designing ways that you can culture cells and coax them to grow in different regions by eroding channels that can be decorated with signals that either do or do not allow certain cell types to follow these pathways and reach a target over here. So this is sort of maybe the future of how complex systems and in vitro culture systems might help us learn about designing better strategies for more complex tissues and problems. But the summary is really that I hope I've given you some examples that hydrogels can be a very versatile environment for delivering cells, for culturing them. They can be designed to be either promoting or permissive, depending upon the desired function and manipulation that you'd like to have. And we walked through some different examples about how you can control gel structure, the role of biophysical versus biochemical cues, how you can design systems that could promote infiltration of cells, and really just advanced biomaterial systems and where the future might be going and what's possible. So I'd certainly like to acknowledge a great group of students, postdocs, visiting scientists. I've had the pleasure to serve as a mentor. All the experiments that I showed you today were the result of their hard work and efforts, and I feel really fortunate to be a faculty member. Specifically, I tried to highlight some of the people's work that I talked about from my group, but we collaborate with Leslie Lindwan on the heart valve project, Chris Bowman on the thiolene chemistry, the peptide degradable materials, and Jennifer Alicia has been a longtime friend and collaborator on the cartilage tissue engineering. And with that, I'd be happy to answer questions.
Thank you, Kristi. That was terrific. So we have time for some questions.
So when you're doing the photo-induced chemistry, you have gradients in light intensity, which must lead to gradients in composition. So how does that limit you, the existence of those gradients, and how do you get around them if you're motivated to?
Yeah, so in general, so a lot of these hydrogels, they have large amounts of water in them, and the concentration of these molecules that are the light absorber is really quite small, like much smaller than you would use for a typical photopolymerization reaction. So if you aren't using 2 photons or lasers and you're using single photon, typically the conditions we work with in the wavelengths, you can get about 90% of the light through about a 2 millimeter thick sample. But if you wanted to have something that's several millimeters or a centimeter thick, then we usually go with strategies where you go layer by layer to create and modify. Yeah.
Other questions for Kristi? Bruce again? Carrie.
Well, Kristi, this was a magnificent presentation covering so many different areas and so elegantly presented. I was fascinated by the work that shows that how you can change I guess the modulus of the substrate and how that modulus can drive so many processes. So I wonder, so that modulus, that stiffness is basically some elastic property, correct, of the material. Have we looked at the time dependency of that stiffness, basically the viscoelastic nature of that substrate, and how might that, that fourth dimension basically in the stiffness?
Yeah, that's a great question. So most of these covalently bound gels, when you look at their viscous modulus, it's very, very small, and they're highly elastic materials. Even when you strain them to large amounts, they snap back. So that doesn't necessarily capture what many native tissues look like. And so one of the more recent things that we're doing is we like the covalent materials because of the range of modulus you can achieve, but we've been working with some chemistries where the bonds are dynamically reversible, and those materials behave very viscoelastic depending upon the frequency. So I'd love to talk with you more about that. So it's been, that's something that we just published on about a month ago. So.
I too enjoyed the talk, and I was really intrigued by the reversibility when you changed the modulus, lowered the modulus, and I guess it was myofibroblasts. But so obviously they mechanosense that environment. And have you looked at the molecules that are associated with the mechanosensing? Can you measure the mechanosensing in real time? And does it drive, or is it a consequence of the changes?
Yeah, so what we have done is we've looked at experiments like right after you've in situ softened, and then we've sort of done data mining of gene expression profiles, and we've shown that the AKT pathway is one of the targets that's really important, and we've looked at inhibiting and overexpressing that, so that's clearly involved. But I think there's more to the story than that. There was some, I guess it's been about a year and a half ago, we're also trying to look at some of the transcription factors in the nucleus that link to the actin cytoskeleton, so some of the YAP and TAZ transcription factors, and that we're studying in real time, and what happens to that as you soften. And finally, the third direction we're going is we're designing systems where they don't just soften, but they soften with one wavelength and stiffen, because we think some of that will be really interesting to go both ways. But yeah, I think right now that we're at a stage where we really have to better understand what's the sensor on the outside and how does that information get transferred all the way to the nucleus to influence these. And so we're just starting to tease some of it apart, but I don't think the story's whole.
So if you look at natural regeneration of cartilage, how does that happen, and what's going on that's essentially different from what goes on in your artificial systems with the dilute, everything dilute in a hydrogel?
Yeah, so I mean cartilage regeneration is fairly limited, and you worry about, am I forming articular cartilage, or am I forming cartilage that's going to eventually turn into bone, right? So it's a lot harder to regenerate cartilage if we use bone marrow precursors versus using primary chondrocytes that we already have isolated from our articular joint. And I think the problem is not that the cells aren't capable of regenerating and growing cartilage. It's just that there's no mechanism once you have a defect created in cartilage for the cells to migrate and infiltrate and regenerate that tissue. So I think fundamentally what we're doing, maybe there isn't a physiologic equivalent to that, but we're trying to recapitulate the density of the cells that would naturally be in that volume of tissue. Yeah.
Are there any organisms that are much better than humans in regenerating cartilage?
I mean, yes. I mean, there are newts and other animals that can regenerate entire limbs, and so I think we just have a lot more specialized cells, I think, in our bodies as well, and aren't.
I was wondering, since you were doing all the gene patterning, have you seen evidence of conditioning or memory so that cells that started stiff and got soft are different than cells that were soft all along?
Yeah. Yeah. So what? So I guess a couple weeks ago we had a paper come out with the bone marrow MSCs that what we can show is that if you passage cells on tissue culture plasticware, if you passage them for 1, 2, 3, 4 times, and then start experiments putting them in soft environments, you've changed the population, and that there's some type of memory of past environments that go beyond. So something irreversible has happened in the cell as a result of being exposed to that matrix condition. And sometimes, some cells, there is a plasticity and reversibility, but for many cells, we see what happens is that it's kind of an integrated over the type of environment that they've been exposed to. Much like when you expose cells to, right, stem cells to the cocktail of, exposed to this growth factor for 3 days and then switch to this one. I think there's something similar going on with microenvironmental signaling. Yeah.
Time for one more question, Carrie?
What, if there
Are there other questions before we go back to Carrie? Carrie?
So the surface modifying technologies, like I said, I find them fascinating. So what do you envision their role in a translational, in an in vivo, in an in situ environment? I mean, in terms of medical devices and instruments, what do you envision one might be able to do with these technologies for medical devices and instruments?
You mean the dynamic
Yes.
system? Yeah. So I think what it allows you to do is to ask lots of questions about, should a signal persist for a certain amount of time before it's released? Should I have sequential signals? And basically, it's one chemistry that allows you to test all these hypotheses about when a signal should go away, when it should be added in. And then I think once you learn that, then now you can engineer the system, whether it's hydrolysis or that will present that signal for that certain amount of time. And so that's, I think, for the practical translation in that case. You might envision certain examples like delivery to the eye, where you could pretty easily have some defined systems that rather than having injections into your eye once a week for some of these drugs, that you could have depots in there that you could then go have a dose of visible light that would then release some of that. Yeah. Yeah.
Well, please join me in thanking Kristi.